DH5 BL21, top 10, XL1-Blue, BL21 DE3. Sometimes it feels like you're living in a DH5 alphabet of E. coli strain names of these different strains of E. coli that we use in the lab in order to do things for us. And there's a ton of different strains, but they're actually just variations of a couple main ones. And we can kind of classify things into cloning cells and expression cells. So cloning cells are really great for making lots of copies of DNA and doing it with high fidelity, so not making a lot of errors, protecting the DNA. And so that's kind of like your first stop when you're doing molecular cloning, is you go into those cloning cells. And that's things like your DH5 alpha. And then you use those to make lots of copies of the DNA. But then if you want to make protein, you got to take it out of those cells, check that it's actually the right sequence, and then put that plasmid into expression cells, something like BL21 or BL21 DE3, if you want to do inducible expression of the T7 RNA polymerase to get inducible expression of your protein that's under the control of the T7 promoter. And so typically when we're doing recombinant protein expression, we're trying to get, say, bacteria to make a protein for us. First, we'll kind of create a plasmid, so a circular piece of DNA that has the instructions for making our protein. We'll stick that into cloning cells, something like our DH5 alpha, that'll make a lot of copies. Those cells are really great because they've got things like nuclease mutations so that they're not going to chew up the DNA, and they have mutations so that it's easier for them to take in the DNA. They'll make a lot of copies of that. And then we can purify out those copies, check that the sequence is correct, and then we stick it into expression cells, something like your BL21 DE3 cells. And those cells, it's not as great at keeping your DNA, like the correct sequence. I mean, it's okay, but it's not like something you'd want to keep it in long term. But it's really great at making protein and protecting the protein that gets made. So it has mutations in genes for like proteases that would chew up your protein. So these cells are really good at blocking those proteins from chewing up the protein that you're making. And those are just a couple of the different types of mutations that are present in these cells that make them really great for the things that we want them to do. And then you have variations of these different strains. And so let's go into some of the features of these different strains. But again, keep in mind that you kind of cloning cells first, then expression cells, and keep those in mind. And then we can go and think about what do you want for one versus what do you want from the other. And then do you need special things? Is your protein toxic to the cell? Does it have rare codons? And so then you might want to consider some of the fancier variations of these strains that, again, just kind of like introducing different things. So some of them have, say, an extra plasmid in them with instructions for the tRNAs for rare codons. Or they have things like extra mutations so that they're not going to chew up your protein. And I mean, like, if it's really, your protein's really sensitive or something. And so a lot of times just the good old basic works, but it's important to know what's in each strain and whether it's compatible for your purposes. So let's dive in. Let's start with the cloning cells because that's typically what you start with in your workflow. You get your plasmid, you stick it into those cloning cells. The cloning cells will make lots of copies of it. You're going to purify out those copies, such as with the miniprep, make sure the sequence is okay, and then you're going to transform it into expression cells to make your protein. The common cloning cells include DH5 alpha, DH10 or top 10, XL1-Blue. There are various proprietary names of different strains. The key features of these are that they're easy to transform. They have inactivated recombinases to keep your DNA from recombining, and inactivated nucleases to keep your DNA from being chewed up. But let's go a little deeper into the different features that actually make it so that they have those key features. This is a lot of text, so let's go one by one. Starting with the name, DH5 alpha. The DH stands for the name of its isolator, Douglas Hanahan. And the alpha, well, that refers to it having something called the alpha complementation allele, which allows for blue-white screening. Don't worry, we'll get into that in a minute. And a lot of the times you don't even worry about the blue-white screening, but know that that is where the alpha comes from, and we will talk about that. It's derived from a K12 strain and has key mutations that make it good for cloning. So what are some of these key mutations? One is that it makes it easy so that the DNA can actually get in there. We call it transformation when the DNA actually gets into the cell, and this REL-A1 mutation is going to alter the membrane composition so that it's easier for the DNA to get into the cell. and it kind of keeps the the cell from freaking out. The cell typically has the ability to stringently respond to low amino acid levels. So basically, when there are low levels of amino acids, which the cells need to make proteins, it halts its RNA production so that it doesn't make protein. However, the mutation allows it to keep making that RNA, even if it can't make protein from it yet, which is good. The DEOR mutation also helps with transformation. So basically, these mutations are going to help your DNA get into the cell. Once that DNA is into the cell, we want to keep it the way we put it in there. And so REC-A1 has a mutation in this gene for this protein REC-A. And that REC-A protein is needed for some types of homologous recombination. And so with homologous recombination, the cells basically say, this region of DNA on this chromosome or whatever looks like this region over here. So let's just kind of like swap the pieces. And that's really important for getting things like genetic diversity inside of our cells. And it's important for some laboratory techniques, but it is not what we want going on now. We want our DNA to maintain its form that we have it in right now, which we might have painstakingly made this cloning. So we want to keep this high fidelity copying, not make mistakes. And so we want to have this REC-A1 mutation. We also want to chew up the DNA we put in there. And so this NDA1, this has a mutation in this gene for endonuclease 1. Endonuclease 1 is a non-specific DNA chewer. And so basically it'll just chew up DNA. And we don't want that. And so this mutation is going to make it so that the cells don't just chew up our DNA. Great. Now, there are other ways that cells can chew up DNA, and so this is another mutation or set of mutations that allows it to keep methylating its DNA and not chewing up DNA. And so basically methylation is the cells, when they're copying their DNA, they have this damn methyltransferase, which will put methyl groups onto the DNA as it's copying it. And this then differentiates between the newly copied DNA and the older DNA. In the lab, this has significance because that newly copied DNA is going to have that methylation. So if you have DNA that was copied inside of bacteria, it's going to have this methylation. But if you have DNA that was copied in the lab in something like a PCR machine, it's not going to be methylated. And this is going to allow you to differentiate between DNA that was copied in the bacteria and DNA that was copied in like your PCR reaction. And that is important because we're often using these PCR-based methods in order to do sort of like site-directed mutagenesis and things and various molecular cloning things. And so basically we take a plasmid that was made in bacteria, you had the copies made in bacteria, so it's going to be methylated, and then you make changes to it and stick it back in bacteria. The way that you kind of select for bacteria that have the plasmid is with an antibacterial resistance gene that's also on the plasmid. But the antibiotic resistance gene is also in the plasmid of the template. And so in order to make sure that we don't just stick the original template back in the cells, the original unmutated or whatever version, we need to chew up that original version we started with. And the way that we can chew it up is by taking advantage of the fact that it's methylated, whereas the PCR produced ones aren't. And so there's this enzyme, DPN1, that we can add in the lab to chew up that methylated DNA. And because those DH5α cells still have that damn methyltransferase intact, they'll have that methylation of their DNA. And since we purified the plasmid out of that bacteria, it'll have the methylation, but the kind of new version we make won't have methylation. It won't get chewed up by this DPN1. And so now when we stick this into bacteria, we're going to be sticking in the mutated version. And now only the bacteria that have that mutated version should be able to grow. The only way to know for sure whether your cloning was actually successful is to go and sequence the plasmid, purify out the plasmid and sequence it. And so typically you take some of the plasmid, you grow like a 5 mil overnight culture of it to get lots of copies of the bacteria so you can get lots of copies of the plasmid. You use a kit like a miniprep kit in order to purify out the plasmid, and then you can do things like send it for sequencing to make sure that the sequence is correct. However, there are also ways that you can kind of get a look at what colonies might actually contain the plasmid that you want. And one of the ways that you could do this is with blue-white screening. Unlike selection, where you're kind of only allowing things to grow if they have some feature like an antibiotic resistance gene, with screening, you're just kind of saying, okay, show me things. So don't get rid of anything. Everything can grow, but if it worked, change color or something like this. And so how it works is the alpha in DH5 alpha tells you that it has this alpha complementation allele. When combined with an alpha peptide, that will form a functional beta-galactosidase enzyme that can convert an invisible compound into a blue one. And so things will look blue if you have these come together. You can design your cloning strategy so that you have your insert, so say the gene for the instructions for making your protein, you put that in a way that disrupts the alpha peptide gene in your vector. Now, if you stick your vector in and it has the mutation, you're not going to make the alpha peptide gene, and so you're not going to make the functional BGAL, and then you're not going to get blue colonies. But if the clone didn't work, now the alpha peptide gene is going to be intact, and so you're going to have your colonies are going to be able to turn blue. And the different strategies you can use, so sometimes you want to look for like the white colonies, sometimes you want to look for the blue colonies. It all depends on your cloning strategy. Most of the time, I don't even worry about blue-white screening, but you can't do that with these DH5 alpha cells, and the alpha is kind of telling you that. So those were our cloning cells, and again, that would be like your DH5 alpha, your DH10, this is also top 10, XL1-Blue. The key features, easy to transform, they have inactivated recombinases and inactivated nucleases. So you take your DNA that you do some molecular cloning, you have a plasmid, you stick that into these cloning cells. You let the cloning cells take in the plasmid. The ones that take in the plasmid are then going to grow as these loopy dots called colonies. In order to get enough of the plasmid for us to sequence and work with, we take a little bit of the colony. We take like a toothpick or a pipette tip, dip it in the colony, stick it in like 5 mils of LV media, grow it overnight. Now you can mini prep it, purify out the plasmid, send it for sequencing. If you want to get a quick look if it might have worked, you can use blue-white screening in some cases, but again, you have to have a plasmid that's kind of compatible with that, and you need to have one of these alpha strains. You can also do techniques like analytical digest and colony PCR, but those aren't going to tell you if the whole sequence is correct, but they can give you an idea as if you to whether a colony is kind of worth sequencing, whether your plasmid is worth sequencing. Because you have a bunch of different colonies, and each of those is going to be kind of like genetically identical to itself, but it might be different from one another, and so you typically test several. Once you know that you've got a good colony, well, now it's time to transform it into expression cells so that we can make some protein. The cloning cells were really great at making DNA and keeping it stable, but the expression cells are going to be really great at making protein. Now, you wouldn't want to use the expression cells to do the copying of the DNA because the expression cells are optimized for protecting the protein, not for protecting the DNA. And they probably won't even make as much of the DNA. And so you basically are going to want to use the cloning cells for the molecular cloning step, and then the expression cells for the protein expression. Yes, you can purify the plasmid out of the expression cells, but be very cautious because there's not as much kind of stability of the DNA, and so the DNA is going to be more stable inside of those cloning cells, and you typically are going to have to make glycerol stocks for long-time storage. So let's talk about what's in these expression cells. And so first off, what are we talking about? So this is going to be like your BL21, your BL21 DE3. We'll get into the distinction between those, K12 and origami strains. When we were looking at the cloning cells, the key things were kind of inactivation of nucleases. When it comes to the expression cells, now we really want to inactivate the proteases, the protein chewers. And so these expression cells have inactivated proteases. And then the BL21 DE3 cells, that DE3, tells you that it has T7 RNA polymerase under the control of the lac operon. And this is going to allow you to inducibly express genes that are under the T7 promoter. Don't worry, we'll go into this in detail. There are also other versions with variants with features like genes for extra tRNAs if you have rare codons, inactivation of additional proteases. There's strains called like P-lysis, which inhibits early T7 activation, various things like this, depending on the specific needs of your cell, like your protein. So if your protein's toxic, say, you don't want to have too early of expression, so you might want this P-lysis. If you have rare codons in your sequence, you might want one of the strains with the rare codon plasmids, and so things like this. But the basic workhorse is this BL21. And so the basic version has these mutations that knock out proteases that can chew up your protein. And then it has mutations in the restriction methylation system to keep nucleases from chewing up your plasmid. So yeah, you still want to basically protect the plasmid, but now it's less about the sequence integrity and it's more about just don't chew it up because I want to make protein from it. And then don't chew up my protein. Although sometimes you'll see BL21 strains alone, often what you're going to be seeing is BL21-DE3. You need the DE3 if you want your cells to make the protein under control of T7 RNA polymerase. And so basically, there's this lac operon, and the lac operon is going to allow you to controllably make proteins on demand. More on this elsewhere, but basically you have your gene with the instructions for making a protein. And then in front of that, you have a lac promoter. And the lac promoter is going to be like, hey, RNA polymerase, bind here and start making RNA. And then the ribosome can take that RNA and make protein from it. It's instructions. But there's a repressor protein that sits on that lac promoter until you induce it. And so basically you can add a compound called IPTG, or if you're using autoinduction, you can basically have the conditions where you're going to run out of glucose in the presence of lactose. Those are going to then cause the repressor to fall off. And when the repressor falls off, RNA polymerase binds, you make RNA, and then you make protein. And you're doing that on demand using this lac promoter. Now, what you could do is you could stick T7 RNA polymerase under the control of the lac promoter. And this is what happens in those cells with the DE3. In the DE3 cassette, basically it has this lac operon controlling T7 RNA polymerase. And so you make the T7 RNA polymerase on demand. Now, we've been talking about T7 RNA polymerase, but what about the protein that we actually want to make? Well, we put that protein under the control of the T7 polymerase. So if our protein is under the control of the T7 promoter, now the T7 RNA polymerase is going to basically be the one that makes the copies of the RNA copies that protein can get made from. But T7 RNA polymerase is only going to get made if you have the T7 polymerase induced. And so basically in these DE3 strains, you induce the production of the T7 RNA polymerase, and then that T7 RNA polymerase is then going to go and make RNA for the protein that you want to make, and then you'll make protein from it. If your cell is under the control of the T7 RNA polymerase, you need to have cells that actually make T7 RNA polymerase. And so the basic BL21 cells won't, but the BL21 DE3 strains do. Now, sometimes you do have plasmids where your gene is under the control directly of the lac operon, and in that case, you don't need the DE3. But if you're under T7 control, then you do. And sometimes you'll have a combination of the two. But it's important to know that the basic BL21 strain does not have the T7 RNA polymerase, and that's actually kind of one of the benefits of using the T7 RNA polymerase is that you don't have to compete with the cell's own messenger RNA making and things. You can kind of have that T7 polymerase all to yourself, but you need to get the cells to make it, and you can have them make it on command by taking advantage of the lac operon and its control of T7 RNA polymerase production in these BL21 DE3 strains. Sometimes you have a protein that's kind of toxic to the cell, and you don't want there to be even like a low level of expression until you're really, really ready. And so there are things like P-lyse cells, and so this expresses a low level of a T7 lysosome that binds to T7 polymerase and kind of inhibits it. And once you have a lot of the T7 polymerase, it's not a big deal, but if you have a little of it, it'll be enough to inhibit it. And so that's going to prevent you from having kind of early expression. Sometimes the problem is that your protein isn't expressing at all. And so this can happen if your protein has spellings that the bacteria is not used to. And so basically the genetic code is degenerate, which means that there are a lot of different codons for some amino acids, and that all of them are kind of spelling for the same thing. So like gray can be spelled with an E or with an A. And both of those are referring to the same color. But cells are going to kind of stock up on the tRNAs for those spellings that they're used to. So cells might have more of the tRNAs corresponding to some codons and other codons. And if your gene has more of the spellings that the cells aren't used to, they'll have less of the tRNAs for using those. And so there are strains of these expression cells, things like Rosetta cells, RIPL, REL, RP cells. These have additional plasmids in them that have the genetic instructions for making those tRNAs for those rare codons. And typically these are going to be on a separate plasmid in the bacterial genome, and that plasmid is going to have its own antibiotic resistance gene. And so typically when you're using one of these strains of cells, you're going to have to add a second antibiotic in order to make sure that you're actually selecting for cells with both plasmids. There are also additional fancy cells. So there's strains like origami strains. This is from the K12 strain of E. coli. This has a mutation in thyrodoxin reductase and glutathione reductase genes. Basically, if your protein has cysteine disulfide bonds, so the amino acid cysteine, it can kind of oxidize and form these crosslinks between regions of proteins or between different proteins. And sometimes this is bad, and sometimes this is what you want. And if it's what you want, then you might want these types of cells that have the inactivation of these reducing proteins to prevent the crosslinks from being broken up. But there you go, cloning versus expression cells. So first you get your plasmid, you transform it into the cloning cells, you make lots of copies, purify out the copies, check the sequence, transform them into expression cells and express your protein, and now you can purify out the protein and play with it. The cloning cells are really great for making lots of copies of DNA and doing it well with high fidelity, not making errors, and not chewing up that DNA. If you want protein, though, you're going to want to stick those into expression cells. These expression cells are good at protecting the protein that gets made, making protein on demand, various things that come with different strains, depending on the features that are most important for you. Speaking of features that are important to you, a lot of the features that you really care about are actually coming from the plasmid with the instructions for the protein rather than the cell strain itself. And so you're going to want to keep a careful look, choose your vector plasmid carefully, things like copy number, selection markers, protease cleavage sites, and fusion proteins. And there's a lot of great information on the web. And also make sure that your vector is compatible with the cell strain that you're using. Best of luck. Hope this helped.